Good morning. I'm pleased to be invited by the organizers to speak to you today about mutated tumor suppressor genes and inherited skin tumors. I'm Neil Rajan. I'm a dermatologist based in Newcastle, and I'll highlight today a little bit about this area and some of the work from my lab. We have been involved in a range of different projects that relate to the genetic dissection of rare inherited skin tumor syndromes. And a key commonality to those is the lack of, sorry, not the lack of, is the presence of a tumor suppressor gene that runs in these families that underlie these conditions. So tumor suppressor genes, or TSGs here, are the main focus of the talk today. I'd like to highlight a bit around the two-hit hypothesis that Knudsen has put forward in the literature and is now a seminal part of this field. I will go on to exemplify this in a particular inherited tumor syndrome, Silkcutaneous syndrome, and finally finish with mosaic presentations. There are learning outcomes that relate to today's talk, and these are the three key learning outcomes, which we'll come back to at the end. But broadly, we would like you to see how a tumor suppressor gene can run in families and play out when a second hit occurs, that these often have a dominant pattern of inheritance in a family that's predisposed to cancer. And sometimes if these second hits happen early in development, you get a different presentation to what you might get if they happen in postnatal life. So maybe the most famous tumor suppressor gene is the breast cancer tumor suppressor gene, or BRCA. And this article in Time magazine a while back had a huge effect on bringing awareness about cancer predisposition to the public with a focus on breast cancer. And this resulted in an increased awareness in testing and also risk reduction mastectomy in predisposed individuals. And I guess no talk on tumor suppressor genes could start without a nod to BRCA. But also as important perhaps is this man whose work on retinoblastoma, a rare type of eye cancer, led to some of the fundamental concepts around the two-hit hypothesis. And Knudsen was someone who had observed the frequency of the development of retinoblastoma in predisposed individuals, and from that arrived at a suggestion that there may be the need for two separate alleles to be individually damaged or mutated such that a tumor would present. And so that's a key part to take away from this talk. So what are tumor suppressor genes? These are elements of DNA that are in normal cells working to regulate processes that are part of normal life and are required for cell division and tissue maintenance. Now, when there is a mutation or a pathogenic variant in one of these genes, the scenario is one where there is a cell process which would normally proceed normally, which can now go unchecked. And this can lead to cellular proliferation, or another route where tumor cells are then given a survival advantage over surrounding normal cells. So if we look at this in the context of sporadic cancer, we have a scenario where most individuals who are not in a cancer family will have two normal copies of any tumor suppressor gene. And over the course of life, through the natural processes that lead to mutation acquisition, one of those copies can become damaged, and that's indicated here in red. Now, in this state, there isn't enough there to allow those cells to go ahead and form a cancer. However, when they lose the second copy, this is the moment at which the cells now escape the normal regulatory mechanisms and then go on to form a tumor. Now, individuals who carry rare mutations or pathogenic variants in certain tumor suppressor genes start life with a slight disadvantage in that they already carry one damaged copy. And so when those normal processes that happen to the rest of us apply to these individuals, they hit this time point when they get their second change sooner. And so if a person has such a change, cancer happens earlier, and cancer happens at multiple cells. And so patients may unusually present with more than one tumor. Now, to go from BRCA to the skin, we have a range of different genetic conditions where mutations which result in second hits can give rise to a range of different tumor syndromes, and these tumor syndromes can manifest in different subcompartments of the skin. And over the last 30 years with the Human Genome Project and a range of other genetic studies, many of these conditions have had a molecular solution. And so the underlying tumor suppressor gene that drives these conditions is now known. The interesting thing is that these conditions are due to genes which have a range and a diverse range of functions. And some of these functions range from elements that relate to the cell cycle, to elements that relate to post-translational modification or DNA repair. And some are obvious as to why they might lead to the formation of cancer. And some are still quite elusive. And so it's an area of great interest. And the skin is often portrayed in the literature as a homogeneous mass of barrier that exists there with hair follicles and the ability to sweat. But here is a retro model of the skin that I quite like because it highlights each of these individual elements. And I see it as a rich tapestry of stem cells that are there to maintain our normal function. However, each one has the ability to acquire mutations and go on to form tumors, which are specific to these structures in the skin. And so to map back some of the conditions that we saw earlier, we have a range of different skin patterns of presentation, a range of different tissues from which they arise from, and a range of different genes which are compartment specific. So we get mutations in some genes that only appear to affect some cells with the predisposition to tumor formation, even though those mutations are in the heterozygous state in all those cells in the skin. So I'm going to highlight now one of these conditions, Sulcutaneous syndrome, which can serve as an exemplar as to how tumor suppressor genes play out in skin. So this condition is not well known, and I'll say a little bit about its phenotype and natural history. Cylindromas and spiroadenomas are terms ascribed to two types of benign tumors that sit in the skin, and the cylindroma is the pink nodule you see here on one side of your slide, and the spiroadenoma is the painful darker blue nodule that's shown at surgery on the other side. These tumors can grow in an endophytic or an exophytic way, and this is not fully understood as to why tumors choose to do these different routes of growth. The patients with these mutations can also develop a third type of skin hair follicle tumor called trichoepithelioma, and these tumors often present on the central face, unlike the cylindromas and spiroadenomas that present on the torso and on the scalp. In addition, they can get smaller changes, which are called milia, which are small keratin cysts that are also thought to arise from the hair follicle. So I've been involved in tumor mapping of these patients previously with a colleague of mine who's now a retired Mohs surgeon called James Langtree, who helped identify the locations of these different tumors in the skin. And interestingly, even though these tumors arise from hair follicles, not all hair follicles are equally predisposed. And so there are tumors at sun-protected sites, for example, that develop tumors. And this is interesting because it challenges existing paradigms, such as UV driving cutaneous tumorigenesis. And conditions like this make us think about expanding that model to recognize that other elements may be important in human skin tumor formation. There is also a female predisposition in this syndrome, and that is a subject of current study. These patients can also develop tumors at sites such as the vulva, which is widely under-recognized, and tumors in the ear, and these can result in conductive deafness unless surgical procedures are done to control these tumors. Interestingly, these patients are born with completely normal skin and over the course of time develop multiple primary tumors. These typically start from the age of 16 onwards, but can start as early as 8 and go on to grow progressively, such that 1 in 4 of these patients undergo complete scalp excision. Sarah Fennick in my lab previously, now a Mohs surgeon in Newcastle, did some work to track cylindromas on CT images over time, and she showed that these tumors progressively grow, and this is seen in the majority of tumors, despite the label of them being benign. And so there is a need to review the perception of this in the literature, because we can otherwise miss an opportunity to treat these patients when their disease is less severe. In different genetic backgrounds, the same condition can present in different ways. So we have a gene that is now quite well characterized, and there are more than 120 families reported internationally. However, families carrying the same mutation who are from different backgrounds from the planet have different sites of tumor formation. So the development of tumors on the scalp, which I've shown you earlier, seems to be largely restricted to families from Western Europe. Gene discovery is often a key moment of excitement for any condition because families feel that they've now got something that will lead to a treatment. And it's important to highlight that in genetics, this hasn't always been the case. And we're always striving to take that initial finding and make it translationally relevant for patients. When the gene for this condition was discovered, John Byrne and Alan Ashworth were involved in the mapping of two large Newcastle families. And these are dominantly inherited traits, as you can see from these pedigrees. Males and females are equally affected. And the genotyping allowed us to confirm the pathogenic variant in each family and allowed us to recognize that the two families in Newcastle were not actually related. They then went on to show that this gene was important in the negative regulation of NF-kappa B and was relevant as a ubiquitin hydrolase enzyme. And the patients who have had mutations in this gene, here indicated with these lollipop-like figures, have had truncating mutations predominantly that are clustering to the 3' end of the gene where the catalytic domain encoding exons sit. And the discovery of this genetic change was helpful because it allowed confirmation that 3 different phenotypes previously reported in the literature were actually caused by the same gene. And excitingly, when we looked across even a single family, now with genotype data, we could go on and say that the 3 different phenotypes that we're seeing were simply not due to changes in the gene itself, but the genetic background in which the gene was presenting. In fact, we then went on to compare the same genetic mutation between a family in Newcastle and a family in Hungary with Niki Nagy, who's based at Szeged University. And we showed that the patients there had a very severe phenotype and the patients in Newcastle had a very mild phenotype, despite carrying the same genetic change in SILD. And recently, Niki has published work on how different modifier genes are now being highlighted that might influence the severity of the phenotype. So to summarize, from a genetic perspective, the data that we've got on this tumor suppressor gene is useful for families because if we can confirm it in a family, it's useful for genetic counseling. A positive test in an affected person is not useful prognostically because this cannot predict how severely an individual might be affected over time. However, a negative test in an unaffected person who wants to start a family is clinically useful because that allows them to go forward knowing that they will not transmit the gene that is mutated down their family line. And so there is still an unmet need to delineate markers that improve prognostication with the aim of early preemptive treatment in those at a risk of a severe phenotype. Now, I've spoken so far about second hits that happen in the skin after birth and go on to tumor formation. However, what about second hits that happen during early development? So there are some individuals who develop a cluster of tumors, and this is not unique to Silkcutaneous syndrome. In fact, in squamous cell carcinoma, there are cases where patients develop multiple squamous cell carcinomas within a birthmark. This is some work that my trainee Anna Dubois has highlighted in Newcastle. And in this work, which was a collaboration with Veronica Kinsler's lab, Dr. Polabothu and Dr. Mathia have both been involved in research that's highlighted patients who develop melanomas only on one side of their body and highlighted that a range of different genes can actually account for these predispositions. And so in Silkcutaneous syndrome, this same model can also play out. So we've seen the unusual presentation of patients who have cylindromas restricted to a small cluster or on one side of their body repeatedly. And in these individuals, sometimes there isn't a genetic test result that is apparent when the blood lymphocyte DNA is checked. And so we have a large national testing center in Newcastle where we've collected DNA from a range of probands in the course of testing over time. And in our mutation positive cases, they have a germline pathogenic variant, as I indicated previously, but there are some who do not have this. And so the 17 in red form that cohort. And so Dr. Arefi in my lab was involved in looking at these cases quite carefully by developing a new assay that sequenced the entire genomic footprint of SILT, so the coding and the non-coding region. And then he applied this to tumor tissue in the skin as well as the blood to look for SILT mutations. Now, interestingly, this showed that in some patients, you could detect mutations in the SIL gene in the tumors that were the same mutation, suggesting that they had come from a similar source, but these were absent in the blood. So these individuals have mosaicism that's only apparent in the skin alone. And interestingly, if you look at normal skin in between some of these two tumors, there are very few reads which are present which carry this mutation, suggesting that these are invisible predisposed patches of skin where these mutations can arise. Now, in some individuals, we see a change that's present in the blood as well as in the tumor, but we may not have detected this on the blood because it was very subtle with prior technologies. However, with next generation sequencing and the ability to sequence at great depth with the assay that Majid Arifi developed, we now have the ability to detect these patients too. Now, in our study, we also had some individuals who had apparently non-clustered or bilateral presentations of their tumors. And this particular case was interesting because when we had sequenced each of these patients' tumors, rather than finding the same mutation in each tumor, which is what we would expect, each tumor had its own private SILD mutation. So this made us consider the possibility that this patient had a large deletion on one allele of the SIL gene, which was the one of interest, and in the remaining allele, that was normal, but would acquire second hits, and then those would lead to tumor formation. Now, interestingly, in this patient, when we checked her blood test, we found that it was normal on an array, suggesting she hadn't got a germline large deletion in SILD. But when we did the array on tumor samples, we found a recurrent contiguous deletion involving 22 genes, including SILD, that covered a 5.5 megabase region. So this information was registered in Decipher, and it was really interesting because this patient had a child who had a single kidney and developmental delay. And when we went on to check the child's germline DNA, we found that she had inherited this in a heterozygous state. So the mum had it in a mosaic form, but the child has it in the germline. And this accounted for her clinical condition. So there are a range of different presentations that you can get with tumor suppressor genes, and the most common is the one seen in the second column, where we see a dominantly inherited pattern in a family. However, you can get early or late second hits that can then influence that presentation. And I would point you to this paper in the Journal of the American Academy of Dermatology to look at this in more detail. In summary, when you see a cluster or linear arrangement of skin tumors, either cylindromas or otherwise, consider the possibility that you might be looking at a mosaic presentation of a tumor suppressor gene, and particularly consider this when germline testing is negative. It's important in these situations to biopsy the skin rather than to do the DNA test on the blood. And it's important to recognize that these changes in the skin can reflect changes in the gonads of the parents that can be transmitted to the next generation. So with that, I will finish with the learning outcomes that we started with at the beginning, and I hope you have found this interesting.